Okay, we're moving on now to the second line of defense. So you've looked at pathogens and antigens, and you've looked at barriers that keep those pathogens out of the body. So now we're moving on to this section here. And you'll see there's a bit to it. So we're going to do this over 2 sessions. So today we're kind of looking at the cellular part, and we'll look at a bit more of it on Friday. So just as a recap, the second line of defense is still part of this innate or non-specific response. Okay, it involves a variety of cells and molecules. You should have all of this written down from last time, so don't write this down again. The aim is to prevent the spread of the pathogen, and it's the same response regardless of the pathogen that has entered the body. Okay, it's the same generalized response that the body goes through. And you'll probably find some of it sounds familiar as we start going through it. Okay, I do want you to take 10 or 15 minutes now to create a poster. This is going to be the immune system overall snapshot. Okay, there is a lot to it, and I don't want you to get overwhelmed because we do spend a few weeks on this, but it's really important that you have this overall summary of what's going on. I know we did a really basic one the other day, just with the first line of defense, second line of defense, third line of defense, but you need to have it a bit more detailed. So please pause this. I've uploaded a PDF copy as well, but it is really low quality, and writing this down will be good. It'll help you kind of get some of those terms in your head. So A4, even A3, do this, have all of this information on there, and you should have this on your wall, for the rest of the year, basically, to help you remember what we go through. Okay, so once you finish that, come back to this and we'll start going through this area here. Okay, so the innate response, a non-specific response, we've looked at barriers already. Today we're going to look at these particular cells, phagocytes and other cells. And then these things we'll look at on Friday. Okay. Again, it doesn't hurt to have this written down, just a quick snapshot of the second line of defense. So this is our non-specific response. This occurs very quickly. So once the barriers have been breached, once the pathogen has entered the body, this response starts within kind of minutes to hours. And the aim is to stop that pathogen spreading around the body. Okay, so a good example, let's say you've cut your hand on like a dirty nail, and there's a tetanus bacterium. So tetanus has gotten into the open wound, the barrier has been breached. Almost immediately your body will launch a response to try and stop that tetanus spreading around your whole body. Okay, the components of the second line of defense, and you'll recognize these from the poster you've just made. We have our cells. Okay, all white blood cells are called leukocytes, but we'll come to that in a second. These are the main cells that the study design says you need to know. If you were to look up a poster of all the immune cells, you'll find there's a lot more. Okay, so we're just going through the key ones. Okay, we also then have what are called non-cellular molecules. So these are really important things in the process, but they're not cells. Okay, and again, you will have written these down, things like interferons, complement proteins, and venom inhibitors. And then it's good to just have an understanding that we have a couple of responses that you'll notice when we go through include a lot of these things, but they are specific responses. So we're going to look at fever and the inflammatory response. We'll look at those on Friday. Okay, but today we're going to look at these cells. Okay, neutrophils, mast cells, macrophages, dendritic cells, and natural killer cells. You need to know each of these and what they do. So, white blood cells, also known as leukocytes. They can be grouped according to their role or their job in the second line of defense. So that's how I'm going to group them here. But you might decide that when you are using the summary poster that you just did, maybe you'll color code things that way so you can group them according to what they do. So we have a group of cells called phagocytes. Okay, and you should remember this term, phagocytosis, is a type of, it involves endocytosis, and it is something where a cell engulfs a part of another cell. You would have looked at that at the start of year 11. Okay, so cells that perform phagocytosis, or the engulfing of foreign or dead materials. Okay, so basically what we're saying here, foreign materials are pathogens. Dead materials might be body cells that, sorry about that line, body cells that are damaged or infected. Okay, the specific cells that are phagocytes, so remember the phagocyte bit, that's their job, that's their role, and these are the actual names of the types of cells. So neutrophils, macrophages, and dendritic cells are all types of phagocyte. Okay, macrophage literally means macro is big, so big eater. Okay, they're just really big cells that eat up foreign or dead materials. Okay, another job, another role that cells can have is called antigen presenting cells. Okay, these are cells that present antigens on their surface, and their job is to activate the third line of defense, and we will come back to that next week. But for now, just have this written down. Remember that antigens can be found either on our own cells or on pathogens. Okay, they have MHC2 markers on them. Remember we talked about some cells have all cells have MHC1 markers except red blood cells, and then certain immune cells also have MHC2 markers. Okay, these are the ones we're talking about. And examples of this are macrophages and dendritic cells. Now you might be going, hang on a second, aren't they phagocytes? Yes. These cells perform more than one job. Okay, so macrophages are phagocytes and antigen presenting cells. Right, the other kind of important cells in this, so they don't, I've put them as other because they're not phagocytes and they're not antigen presenting cells, are natural killer cells. And as the name suggests, they kill things. So they recognize foreign antigens on an infected body cell, and they tell that cell to apoptose. Okay, so let's say this is one of your cells and there's viruses in there. So this virus has infected the cell. Because that's happened, bits of the virus are now on the outside of your cell, and here is a natural killer cell, and it is basically going to send a message to this cell to apoptose. And we'll look at that again in a minute. There's also mast cells. Okay, mast cells are found in the connective tissue around the body, and they release a signal called histamine, which triggers an inflammatory response. Again, we'll come back to that on Friday when we look at the inflammatory response. I know a lot of this is me saying we'll come back to that. We will. We'll tie this all together. Okay, so once you've got that written down, jump in and answer question 5 in the Google quiz. Okay, you do need to know the process of phagocytosis. So we mentioned before that this is when foreign material or waste is engulfed and broken down. And these are the types of cells that do this process. So neutrophils, macrophages, and dendritic cells. I recommend drawing this, or if you're working online, going online and finding a picture of this. Okay, so the first thing that happens is that the phagocyte, which is either a neutrophil or a macrophage or a dendritic cell, engulfs whatever material it's going to break down by endocytosis. Okay, so if you look at this picture, we've got the phagocyte, the cell here, the big blue thing. We've got the pathogen or the waste that needs to be broken down. Okay, so that could be a virus, or it could be, a bit of damaged cell material. And remember, lysosomes are a type of organelle that you would have written down a lot in year 11, but we haven't looked at them a huge amount so far. But we're going to look at them now. So number 1, the phagocyte engulfs that material by endocytosis. When that has entered the cell here, it's now known as a phagosome. Okay, so that's basically it's pretty much a vesicle with the material in it that needs to be broken down. Now that phagosome will actually fuse with a lysosome. Okay, so the lysosome is also kind of like a vesicle, so they will fuse together, and what you're getting now is the enzymes that are in that lysosome are mixing with the material inside the phagosome, which is either a pathogen or a bit of waste. And then the enzymes will act upon that material and break it down. Okay, so you can see here now the blue and the orange are all kind of mixed together because those enzymes have broken down the bits of that pathogen or that waste material. Okay, and then we don't want all this stuff sitting in the cell, so the final step is that the waste material will get released by exocytosis. So the plasma membrane will fold and open and allow that waste material out. Okay, so have something drawn or written down in your notes and then jump back into that quiz and answer question 6. Okay, like I said, we will come back to antigen presenting cells next week, but I do want you to have this written down because it relates to these cells that we were just talking about. So remember, macrophages and dendritic cells are examples of antigen presenting cells. Okay, they have MHC2 markers on their surface. Remember, that's a self-antigen. After an antigen presenting cell breaks down a pathogen, so phagocytosis happens first. Once they have done that, what they do is actually save bits of this foreign antigen and display that on their MHC2 marker. Okay, this is a pretty different diagram to what we just looked at with phagocytosis, but it's the same thing happening. We've got up here, it'd be a pathogen with an antigen on it. So that could be a virus, it could be a piece of a broken down bacterium. Okay, endocytosis occurs here, so the pathogen enters. We have a phagosome there, and then the enzymes break down the antigen and the pathogen into really, really small pieces. Okay, now what is kind of different here, we would still get most of the waste getting removed from the cell, like it would in phagocytosis. But what is special about these cells is they hold on to a few of these pieces of antigen. Okay, so most of it gets released, but some of it they hold on to and they actually put it onto their MHC markers and basically wear them on their own surface. Okay, so they display the antigen on their MHC2 markers, which is the yellow thing. The reason they do that is that they then bring that antigen to a helper T cell, and that will start the third line of defense. Like I said, we will come back to that. So for now, I just want you to have this written and drawn and just understand what an antigen presenting cell is. And that's just a picture there of a macrophage presenting an antigen to a helper T cell. Like I said, we'll come back to that next week. For now, you just need to know that amount of detail and just jump into question 7 and answer that. Okay, I just want to clarify apoptosis and phagocytosis, because this term came up, phagocytosis came up when we looked at apoptosis, and they are related but different. So remember, apoptosis is the programmed death of a cell, and this happens if a cell is damaged, if it's not needed anymore, or if it is infected. And that's probably what we're focusing on mostly here, a cell that has been infected. So let's say a virus has gotten into a cell. Now, natural killer cells, you remember they were one of the cells in the other category that we just looked at. They send death signals to infected cells that initiate apoptosis. Okay, so we've got a cell here, it's got viruses in there, it's infected. Okay, a natural killer cell sends a signal to that cell for it to go through apoptosis, which is what is happening here. Now, apoptosis, remember, we get those caspases are activated, all the things inside start to break down, the membrane starts to bleb until eventually the membrane breaks off and forms apoptotic bodies. And then along comes a phagocyte and engulfs those bodies, which remember now, phagocytosis is when the fragment gets engulfed and you have the phagosome, and then it fuses with the lysosome and everything gets broken down until eventually we're getting these tiny bits of waste get released. Okay, so they're very closely related, but they are two very different processes. For a phagocyte to engulf apoptotic bodies, apoptosis has to happen first. Okay, and I guess the other thing to think of now with the natural killer cell, it sends a death signal. Remember, we had two types of ways that apoptosis could start. It could be a death receptor pathway or a mitochondrial pathway. And we think about which one it would be. If it's coming from this cell, it's coming from a different cell and telling it it needs to destroy itself because it's got viruses inside. Okay, so starting to connect some of the content that we've learned together, and it's really important that you can do that. Okay, so go into questions 8 and 9 and have a go at those. All right, so we looked at barriers last week. Today we've had a look at phagocytes and in particular the neutrophils, mast cells and macrophages and dendritic cells, and also what natural killer cells do. Okay, we're going to move on to these in the next lesson. Okay, so I definitely recommend that you go over this stuff again before we move on. There's going to be a lot of new terms coming up over the next couple of weeks, so the more you can practice those, the better.